Hello, everyone. We're going to just wait a minute for things to get started, for everyone to get let into Zoom. Okay, let's get started, everyone. Well, welcome to the ASCCT ESTIV webinar series. I'm Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. This is our July webinar. I want to encourage you to visit our websites, which you can see on the screen, to find out more about our organization and consider becoming a member, because a lot of the activities that both of our societies do are to support early career and current career scientists in advancing their work in vitro and computational methods. The webinar will be recorded, and the recording will be posted on the ASCCT website a few days from now in our webinar archive. And in the webinar platform, you can ask questions of the presenter using the Q&A feature in the toolbar, which is usually at the bottom of your screen. There will be a question and answer period at the end of the presentation, but feel free to add your questions at any time. And other participants can see your questions and vote on them. We have also enabled closed captions, and you can turn them on or off in the toolbar as you wish. And please say hello in the chat and let us know where you're joining from. So before we get started, just a couple of quick announcements. Our annual meeting is coming up in October. It is going to be in Gaithersburg, Maryland, which is outside of Washington, D.C. at the Institute for In Vitro Sciences. And we have two confirmed plenary speakers, Dr. Nicole Kleinstreuer, who's acting NIH Deputy Director for Program Coordination, Planning and Strategic Initiatives, and Dr. Tracy Beth Hoke, Senior Advisor for Clinical Sciences in the Office of the Commissioner. Both of these wonderful scientists will be giving presentations. We also have the draft program up on the website so you can see the sessions. And we still have abstracts open. You can see the deadlines at the bottom of your screen, so please feel free to submit your abstracts so you can participate in the meeting. The ESTIV Congress will also be in June of next year, so head on over to the website and see where they are with that meeting as well. And for today, I will now introduce our presentation. So we will be hearing about a next generation risk assessment for occupational chemical safety, a real world example with sodium-2-hydroxyethane sulfonate. Our presenters are going to be Adam Wood and Steve Gutsell from Unilever. Let me introduce them briefly. Adam is a safety scientist and toxicologist who has been working in Unilever's safety, environment, and regulatory sciences team in the UK for around 5 years. And prior to this, worked as a toxicologist at Boots, following the completion of his undergraduate and master's in pharmacology at the University of Liverpool. At Unilever, Adam has experience in the safety assessment of many different product types across Unilever's portfolio, such as foods, cosmetics, and cleaning products. And he also works in the development and application of new approach methods as part of Unilever's ongoing efforts related to next generation risk assessment, particularly in the areas of food safety and occupational safety. Steve Gutsell is currently head of regulatory science and chemical safety at Unilever. He gained a BSc in biomolecular and biomedical chemistry from the University of Wales, Swansea, where he went on to obtain his PhD in organic chemistry. Since joining Unilever in 2003, Steve has developed expertise in computational chemistry, specifically using QSAR read across and other techniques to predict both toxicological and ecotoxicological endpoints from chemical structure. He's published many papers in this area and presented at numerous international scientific and regulatory meetings. As well, during his career, Steve has worked closely with colleagues in Unilever's Global Regulatory Affairs team to advocate for the use of non-animal methods in chemical registrations and next-generation risk assessment. Let me invite you to check out our calendar on upcoming events. We have several webinars coming up next year featuring winners of awards from we always have webinars after our meeting featuring awards winners from the annual meeting. So we'll be doing that again next year. But for now, we don't have anything on the calendar, but our partners do have some of these events going on. So feel free to visit the calendar to find out about that. And then I would also encourage you to put your topic or speaker suggestions in the chat if there are particular webinars that you would love to hear about. Okay, I will now turn the floor over to Adam and Steve.
Perfect. Thanks very much, Christy, and welcome to everyone. I've been nicely distracted by the chat, so I think good morning, good afternoon, and good evening is appropriate. I've seen California to Mumbai in the participants, so that's really, really fantastic. Excited to talk to you today about what we've been doing at Unilever in the last couple of years, which really is the culmination of a good couple of decades of work. So we're going to be talking all about next generation risk assessment, and Adam is going to deliver the meat of the presentation. I will pop up from time to time. Think of me as the cheap carbs in the club sandwich of this presentation. But yeah, if you've got any questions, please drop them into the Q&A function and we'll try and leave plenty of time for that at the end. Next slide, please, Adam. So here's a quick rundown of what we're going to talk about. We're going to talk about worker safety, an application of NGRA to worker safety. So a little bit of a view on the current approaches, and then into next generation risk assessment, an external perspective followed by an internal perspective, and a really interesting summary of Unilever's long journey in this space. And then we'll talk about the case study, sodium isethionate to its friends, or SI. It's a real world example linked to a REACH registration that we will get into slightly later on. And then we'll finish with a bit of looking into the future, particularly, forgive us, but with an EU perspective, because that's been a big focus of the case study. Next slide, please, Adam. So in terms of historical worker safety assessments, much like all chemical risk assessments, they have been predominantly based on animal studies to determine safe levels, be they occupational exposure levels or other derived no effect levels. And we know that this practice is outdated, both from a scientific and an ethical point of view. New methods are emerging in various parts of the world, various agencies, various industries, various academic groups that start to tackle chemical risk assessment without that reliance on animal testing. Next slide, please, Adam. So in terms of the opportunities to improve occupational risk assessments, we've already seen big shifts in the last 10 to 15 years in terms of local effects. So things such as skin and eye irritation, sensitization, animal testing is virtually replaced for those endpoints in most parts of the world, not everywhere, but in most parts. But systemic effects have proven much more challenging, including developmental and reproductive effects, really because they require a fundamentally different approach from a one-to-one replacement or a simple defined approach, such as those for sensitization. So some regulations, in addition, have some fairly significant issues in terms of how they're set out. So particularly within Europe, in the REACH regulation, tonnage is used as a surrogate for human exposure, as a pragmatic way to attempt to tier the information requirements. Clearly, this is not a scientific approach at all. And the science behind risk assessment has moved on significantly, and the regulation, frankly, has not. So the question is, how do we bridge that gap now that we have much more science at our fingertips, high throughput non-animal methods, etc.? And the answer to that is what Adam's going to talk to you about over the next few slides.
Okay, so thanks, Steve, and hi, everyone. It's really great to have so many of you on the line today. So like Steve said, over the last several decades now, there's been really tremendous investment made in the development and application of various non-animal methods or new approach methodologies or NAMs, and they can be broadly classified, of course, as in silico, in vitro, and chemico, and they can range in their complexity from quite limited throughput, limited data approaches like reporting cell lines that maybe tell you about one specific biomarker, one specific endpoint, but much more potentially to much more data-rich approaches like transcriptomics, which could tell you maybe many hundreds of thousands of different things. But broadly speaking, we really use these new approach methods in quite a similar paradigm to how we've conducted toxicology and risk assessment historically. So hazard identification, characterization, exposure estimation, and risk characterization. But the key thing is, instead of a margin of safety, so your ratio between your point of departure, your no observed adverse effect level, and your exposure, we're deriving something called a bioactivity exposure ratio, or BER. So our ratio between our point of departure from our NAMs and our exposure estimate as well. And you can see quite nicely down the bottom a screenshot from a Health Canada report from a few years ago, how you might actually use these BERs in a safety decision-making context. So you've got some chemicals at the top with very large bioactivity exposure ratios, but some chemicals down the bottom with very small bioactivity exposure ratios. So those chemicals at the bottom, bioactivity seems to be happening at the same doses or same concentrations as exposure. But one really key thing and one real step change, I think, with next generation risk assessment is the potential tiering to it. So perhaps those chemicals at the bottom, there's still quite a few of them. So from a prioritization or a subsequent decision-making activity perspective, you might want to say run those chemicals in higher throughput or higher data richness or higher tiering models, such as organ-on-chip, etc., from which you can perhaps split those high-risk chemicals into different groups to help prioritization. And broadly speaking, we can kind of classify NAMs into two different kind of conceptual groups. Those are NAMs that are based on the premise of prediction and NAMs that are based on the premise of protection. So NAMs that are based on the premise of prediction are really those that are trying to predict certain types of apical effects, such as, in this example, cardiotoxicity, liver toxicity, kidney toxicity, etc. But you can see already, just from the two organs here, that you're already looking at quite a mind-boggling number of potential assays. If you scale this to, say, the 78 organs that we have as humans, you're looking at a really, really vast battery of assays, which obviously clearly isn't practically feasible, nor do we actually have, of course, predictive NAMs for that, for all of those different kind of organ toxicities. So a more pragmatic approach is, of course, really based around a protective paradigm. So that's the use of more general, broad coverage, high throughput NAMs that give you information on the general likelihood of biological perturbation at consumer or occupationally relevant exposures. And really the state of play or the state of the art has really shifted in the last 20 years or so from these methods, as I said, moving from quite low throughput, low data richness approaches like reporting cell lines through to much more data rich approaches like transcriptomics. But I guess, again, a key message here is that how we might want to use NAMs in the future, probably going to be a mixture of both protective and predictive NAMs, is really quite similar to how we've done risk assessment historically. So combining both broad coverage in vivo studies, like a 90-day subchronic study, but also with mechanism or specific endpoint tests. So, for example, a HERG in vivo study, for example. And really the power of potential protective-based next generation risk assessment has really been nicely emphasized in some fantastic papers by the Accelerating the Pace of Chemical Risk Assessment, or the APCRA initiative, which includes scientists from US EPA, US FDA, European Chemicals Agency, and amongst others. So in this first seminal paper from 2020, they took over 400 chemicals from the ToxCast database, so assessed in many potential hundreds to potentially thousands of different assays, and compared their points of departure from their NAMs with their traditional animal counterparts, and really encouragingly showed that, you know, over 90% of these chemicals in that data set had a point of departure from the NAMs that was more conservative than the pod traditional. So you could potentially use these NAMs in a protective way. But of course, using approaches like ToxCast aren't feasible for the most of us, and the general state of the art has moved on probably from batteries of that size towards data-rich approaches like transcriptomics. And again, this same group of scientists earlier this year, in fact, published their latest evaluation of a new protective battery, so including methods and approaches like cell painting and phenotypic profiling, again finding very similar overall findings in terms of pod NAM being generally conservative of pod traditional. So moving towards how we in Unilever really have translated these principles and methods helping us to develop our confidence with these models, building up our internal best practice, and really allowing us, me and Steve in particular, to go from how we've been applying these NAMs and NGRA to a consumer safety perspective, so assessment of things like cosmetics, foods, etc., to trying it in different sectors, so worker safety, which is obviously why everyone is here today. And we've really done this by moving from case studies on small numbers of chemicals, so which I'll talk about in a second, through to actually building up set batteries of assays, analyzed in set ways, using PBPK models set up in set ways, and really based around both systemic or general toxicity, but also developmental and reproductive toxicity. So our first paper in this area was a case study. So we published this in 2020 now, so a few years ago now. And this really set out our conceptual framework. And visually, you'll see this, the graphics here will be repeated throughout this presentation. But we really start this conceptual framework with a really comprehensive problem formulation. So looking at things like in silico alerts alongside a potentially systematic literature review. So collating all of that existing information. In parallel, we're using a variety of different exposure estimation modules, both at an external level, so potentially in mg per kg or mg per meter cubed for inhalation exposures, for example, before translating those exposures through to internal exposures, so the use of physiologically based kinetic modeling, so parameterized using various standard ADME assays like fraction unbound, hepatic metabolism, etc. Then in terms of the actual NAMs to try and characterize bioactivity, we're conducting several different studies, which include things like transcriptomics, so very data-rich approaches, and in 3 different cell lines, so a HepG2 cell line, a HepRG cell line, which gives us some metabolic competence, and then MCF7 cells. And then we're using a cell stress panel, so looking at things like mitochondrial toxicity, oxidative stress, other kind of very common mechanisms of action for industrial chemicals in particular. And then finally, an in vitro pharmacological profiling battery, so sets of receptors, ion channels, and things like that, which are obviously very common causes of toxicity. So beyond the positive findings of this first NGRA case study, i.e. that we could derive points of departures, we got the same ultimate end risk assessment result as if you actually just used the traditional animal testing data. So really built up our internal confidence. But of course, the open question with case studies is, well, what happens when you start to test more chemicals through it? So over the last few years in particular, many people within Unilever have been working on developing and evaluating a toolbox of different NAMs. So this is a group of NAMs that we will use every single time going forward for a risk assessment in this context. So that's the cell stress panel, your in vitro pharmacological profiling, and your transcriptomics. So it's a toolbox of different NAMs. So our first goal of this paper was really around setting a threshold bioactivity exposure ratio. So essentially, what is a bioactivity exposure ratio above which you would be able to conclude that a substance exposure is a low risk? So we set this bioactivity exposure ratio threshold by testing 10 chemicals with known risk classifications a priori, so either a high risk or a low risk. So example, high-risk chemicals being drugs that were withdrawn from the market, pesticides, etc., and low-risk chemicals being perhaps cosmetic ingredients with opinions by regulatory authorities, ingredients with extensive history of uses, etc. So the output of this first paper was a set of threshold bioactivity exposure ratios. But we then wanted to see, well, are these bioactivity exposure ratios any good when we start testing a much larger group of chemicals in them? So at the end of last year, we published our final evaluation of this toolbox and workflow. So we tested an additional 38 chemicals in this toolbox, and then again, derived bioactivity exposure ratios. And then to actually evaluate whether these thresholds were acceptable or not, we developed these fairly novel metrics called protectiveness, which is simply the percentage of high-risk chemicals that were correctly identified as high risk, and utility, the percentage of low-risk chemicals correctly identified as a low risk. And we got really encouraging results. So over 90% of our high-risk chemicals were correctly classified as an uncertain risk. And utility was a little lower, sort of around 10 to 20%, which really means that you would potentially need to go to higher tiers. But these have mainly been in the area of general systemic toxicity, but what about developmental and reproductive toxicity, right? So a few years ago, some of my colleagues published our first conceptual framework in this area. So again, a framework for how you might conduct next generation risk assessment for DART. So it looks very visually similar, but it's got a couple of unique add-ins to it. So, for example, an additional couple of types of PBPK modeling, so looking at pregnant women as well as modeling the fetus, but then also into the biological activity characterization module, where in addition to your same tools that we're using in the systemic toolbox, so that's our in vitro pharmacological profiling, our cell stress panel, and our transcriptomics, we're also doing a couple of DART-specific NAMs. So that is the Stemmer DevTox Quick Predict and Toxisys Repro Tracker. So both stem cell-based assays. We then published our initial evaluation of this framework literally a few weeks ago, and the references are on the previous slide. I'm happy to post the links to these papers in the chat after I finish speaking. So we published our first evaluation of this framework a couple of weeks ago. So again, we tested a mixture of high and low risk chemicals and derived their bioactivity exposure ratios. And really encouraging, for the vast majority of chemicals, we could separate those high and low risk exposures. So again, really positive news from the potential of making protective safety decisions.
Okay, thanks, Adam. So let's turn our attention to the case study. And this is a real world case study. We, like many other companies in the EU or who operate in the EU, received some letters from the European Chemicals Agency over the last few years. And those letters were in the form of compliance checks on substances that were already on the market, indeed had been on the market for many decades in some cases. And these compliance checks effectively highlighted what were perceived as deficiencies in the REACH registration dossier. Deficiencies in the hazard data that was, or the so-called standard information requirements, these tiered lists of studies that are requested for chemicals that are manufactured or imported into the EU. And when we received those for chemicals that we knew to be safe and we knew to be handled by workers in a low-risk environment and safe indeed for consumers, we wanted to push back on those requests because we really did not see the need for additional animal testing, and that's what was being requested for these substances. So we decided to push back and to use the legal text and the adaptations that are written into the REACH regulation, but are, if we're honest, not often used to their full extent, to see what we could do. And this was around the same time as Adam's just been telling that narrative of our NGRA journey. The request that came through on this particular substance, the sodium isethionate, was around worker exposure because it's exclusively used in cosmetics and the cosmetic exposure is covered by a different regulation. So we decided to turn our NGRA toolbox approach to worker safety. We'd not tried it before, no one had, as far as we were aware. And we instantly came up with some challenges, and they're listed out on this slide here. Simultaneous exposure over multiple routes, different exposure estimation models, external exposure, compared to what we were used to dealing with for consumer cosmetic exposure. Long lists of exposure scenarios in factories. We needed to cover sites that were manufacturing the chemical and sites that were handling it to formulate finished products. And in general, when we talk to our chemical supplier colleagues, the supply chains around some chemicals are very complex, and it's not always clear which chemical is used by who and where. So there is a logistical challenge as well as a scientific challenge. So to address that, we thought we need to jump in with 2 feet. We need to build some case studies, and we were doing it in real time on a real example with other companies who at first were a little reluctant to follow the non-animal approach and to challenge the status quo. But as you'll hopefully see, and we got positive results, they've now become advocates of this approach. On the next slide, you'll see an adaptation of the workflow that Adam has already talked about. So the part on the left is the standard, if you can call it that, NGRA workflow. In doing this on a real live example for worker safety, what we realized was that we needed to progress to higher tiers. As Adam indicated, if you get a BER that appears unfavorable or indicates uncertain risk, that's not the end of the story. There is plenty of scope to refine your risk assessment. And in the case of worker safety, particularly around the occupational exposure estimates, because we are dealing with estimates, not measurements here, there's a huge amount of scope for refinement. And we had to go through a couple of cycles learning some of the new tools to reach a more realistic but still conservative understanding of the exposure. And over the next few slides, Adam will talk you through for SI how we built this risk assessment in a real world case study.
Okay, so thanks, Steve. So yeah, so on to SI then. So just to give a bit of background then in terms of its use. So SI, or sodium-2-hydroxyethane sulfonate, as you would have seen in the webinar invitation, is a widely used ingredient in the manufacture of various widely used surfactants known as alkyl isethionate surfactants. So includes surfactants like sodium cocoyl isethionate, sodium lauryl isethionate, for example. So extensively used surfactants in products such as shampoos, but also potentially cleaning products as well, both by consumers at home, but also potentially by professional cleaners and other workers as well. SI has been the study of a reasonable amount of historical attention, toxicological attention. So it's been subject to a 90-day study and a prenatal developmental tox study, both high quality studies, guideline, GLP compliance studies, for example, in the early 2010s. Across both of those studies, there hasn't been major toxicity observed. In the 90-day study, the no observed adverse effect level was the lowest that exists across the database at 200 mg per kg. But as Steve said, it does have a data gap in the area of a generational reproductive toxicity study, for example. So really, to go around the start of our NGRA, like I said, we start with a comprehensive problem formulation. So that takes into account our historical toxicology data, any other literature data that might exist, but also follows a fairly comprehensive in silico profiling battery using a variety of different tools, some of which are shown on the slide. And the overall trend for SI was, as could be expected, based on its structure and its use patterns and other factors, really no alerts across the board, which gives us a bit of confidence going into the next generation risk assessment. The next stage of our NGRA was probably the most complex from our previous experience, and that was really around actually understanding, ultimately calculating external exposure estimates. So mg per kg from dermal worker scenarios, but mg per meter cubed from an inhalation perspective. And this is really challenging because of the need to conduct very extensive and very complex life cycle assessments that require the integration of a lot of different individuals, but especially in context of a REACH dossier where you're working with multiple registrants, co-registrants, downstream users, can become incredibly complex. So a real learning from me and Steve during this project was the value of expert independent consultants who have real expertise in getting to the bottom of life cycles for complex reach chemicals with complex supply chains. And actually, if you're in the chemical industry and on the line, you'll probably look at the SI life cycle and go, that's not too complex. But we do have much more experience over the last year, not for SI, but for other substances. And there's probably people on the line that have also been part of working with us in consortia that can attest to that we have been able to do these comprehensive life cycles for substances with very complex supply chains. So we first then estimated, we got all of our process categories. So how are workers exposed to SI? What are they doing in factories? Are they professional cleaners? How long are they doing tasks? Is there ventilation? Are they wearing gloves? Do they have masks on? How long are they doing the task for? How long is their shift? All of those kind of factors are taken into account. And from all of that contextual information, we then translated it to external exposures, so mg per kg or mg per meter cubed. And we used a variety of different external exposure estimation modules like TRA, but others are available. And we then use those external exposure estimates as an input to an SI-specific PBPK model. But as Steve said, one of the real complexities with occupational risk assessment is the simultaneous exposure that occurs across both the inhalation and the dermal route, which makes PBPK modeling quite challenging and potentially would mean that you have to conduct an ADME battery that's reflective of the inhalation route and an ADME battery that's reflective of the dermal route, which becomes quite technically challenging. So what we've developed as a first very protective, very conservative tier is the use of a 6-step procedure that essentially can convert your dermal inhalation external exposure estimates into a single IV infusion dose that takes into account everything such as how often workers perform the task, how long they're exposed, things like the skin penetration for SI. So we did a guideline compliant skin penetration study for SI as well to feature into the risk assessment. And then we get at the end an infusion dose that can serve as an input to a PBPK model. So a single dose that takes the complexity out of your inhalation and your external dermal exposure estimate. So goes from 2 to 1. But one of the real uncertainties that we've still got, and a positive uncertainty, it means that we're being quite protective and conservative in our end risk assessment, is that we're assuming while we're refining our dermal exposure for dermal penetration figures, we're not doing anything with our inhalation bioavailability because of the current lack of models in this area. So it's a real call for capability in this area across the field. So we then obviously built our actual PBPK model for SI. So as I said, we conducted a variety of different SI-specific ADME studies, so fraction unbound, hepatic metabolism, skin penetration, for example. And we then built 3 different types of PBPK models. So that's a deterministic model, so a model that's built for, say, a mean average adult within the population, but then moved to 2 more probabilistic models. So probabilistic 1, as I've called it there, where we're essentially just varying the physiological values of this model, so varying things like organ blood flows, for example, and then move to a fully probabilistic model, so that's variable physiological values and variable parameter values for things like potentially uncertain parameters like fraction unbound, for example, if you've got uncertainty in the actual experimental study that you've used. And the end of all of this is a set of either mean or 95th percentile distributions of your Cmax, which we take forward for our risk characterization. So the Cmax for SI, the most conservative Cmax for SI was a figure of 0.8 micromolar. And you can see from this nice concentration time profile that by the end of the shift or by the start of the next shift, the SI has completely been removed from the worker's bloodstream. So there's no accumulation over time. We then tested SI in the same bioactivity studies that we have across our DART and systemic toolbox. So our cell stress panel, our in vitro pharmacological profiling, transcriptomics, analyzed in two different ways, a model that looks at single gene changes and a model that looks at pathway changes, and then finally our DART-specific assays. So that's our 2 stem cell-based assays, our Stemina and our Repro tracker. And as expected, we saw very low levels of bioactivity across the board, very high points of departures relative to what we've been finding in our systemic and DART toolbox, for example. One quite interesting thing that we did as well was that one of the big uncertainties when you're conducting next generation risk assessment is how well do nominal concentrations, so the concentration of a test substance that you put in your wells with your cells to ultimately get a point of departure from, how well do those reflect actual concentrations that are achieved? Maybe you've got things like volatility that's coming into the mix. Maybe you've got binding to plastic, etc. And there's certain phys-chem properties that are really nicely explained in this publication from my colleague Beata, reference here. And again, we can share the reference in the chat. And really, this is a workflow that helps us understand whether a chemical is likely to show potential issues when testing in vitro. So SI doesn't have any of those properties, but we still did some dose confirmation work, and we found some potential actual dosing errors by the company that conducted the assay. And so we corrected the point of departure for the actually achieved concentration in the assay. We then combine these points of departures with our exposure estimates to derive our bioactivity exposure ratio. And you can see here the margin between our lowest overall point of departure, so that from our transcriptomic study, against our Cmax at our 95th percentile, either in our pregnant population or our general workforce population. And we did this pregnant population due to the absence, of course, of a generational reproductive tox study. So our overall lowest bioactivity exposure ratio was 130, but of course, if you were to look at it at distribution level, many of them would be, orders of magnitude higher than that, given the spread in the bioactivity data, but also in the exposure data. So in combination, overall, we concluded that with a bioactivity exposure ratio of 130, along with our lack of in silico alerts, that current occupational exposures to SI were well controlled. And really, actually, this decision is consistent with one that could be made with the use of the current in vivo data set, albeit with an additional uncertainty factor for the absence of a generational reproductive tox study. So when you compare at an external level these same exposure scenarios that we used in our PBPK and that form the basis of our lowest bioactivity exposure ratio, we get risk characterization ratios of much less than 1. So anything less than 0.5 is generally considered to mean that a scenario is well controlled. So both our traditional risk assessment and our next generation risk assessment are consistent with one another. Steve?
Thanks, Adam. So just to wrap up then, what I think, and I hope Adam has convinced you, is that for this particular substance, from a bioactivity point of view, there's really very little going on in that broad range of assays. And that's consistent with the in silico profiling results and also consistent with the existing knowledge on the substance from historical studies. Together with the internal estimates of worker exposure, then we can confidently state that the current occupational exposures and the risk management measures already in place, the good manufacturing procedures, PPE, etc., is sufficient for the protection of workers. And critically, the performance of additional animal testing would not provide any benefit to human health. Or in REACH language, animal testing wouldn't really be the last resort. We've shown that actually using these NAMs and these NGRA methods, we can demonstrate the safe use. And in general, we knew that there was a lack of published examples of NGRA for worker safety, and we'll give you the reference at the end. It's no surprise that we published this. And we think that this framework, especially the tiered nature of it, which gives you options to refine, is actually applicable to probably quite a large subset of substances which need worker exposure and worker safety assessments. It may not be sufficient for all, but it should be for a large number of them. We've also included the simple procedure for converting the external inhalation and dermal exposures to infusion doses to get over that external to internal hurdle that you need to be able to use the PBK modeling and ultimately come up with internal exposure concentrations. And we think that frameworks such as this also could be used to revise how some of the regulations are implemented fundamentally. So moving away from a tonnage-driven surrogate for exposure to a more risk-based approach, using these higher throughput, higher content methods that lend themselves to screening perhaps larger numbers of chemicals. And finally, looking into the future, there's a large activity going on in the EU at the moment with a roadmap just about to be published on phasing out animal testing across all pieces of chemical legislation, including worker safety. And there will be a greater emphasis on non-animal methods in the guidance, in the legislation, including the revision of REACH, which is also expected in the next few months. And from our observations, and when we've been working with our colleagues and our suppliers and across REACH consortia, we've really realised that what's needed is still some considerable work, but it's different work to what we've been used to doing. So rather than defaulting to going to do the standard OECD test guideline animal study, if you can divert some of the resources to understanding the life cycle of the chemicals, the uses, and characterizing the exposure to a higher degree of resolution than we've been used to in the past, then it really does open up the potential for these novel approaches, these non-animal approaches, next generation risk assessment to be used to give confident conclusions about the safe use of chemicals. And even if you do get a screening level result that looks like uncertain risk, as we've talked through, there is plenty of potential because of the conservatism in several parts of the NGRA framework to refine those so that if you do end up with a conclusion that you need to do animal testing, you really can be confident that it is as a last resort, and it should be far less frequent than it is today. And then finally, just to wrap up before we take the Q&A, you can probably tell from the amount of work involved here that it takes a village to do one of these NGRAs. So a large number of people, I'm not going to read them all out, but critically for this example, as well as Unilever safety scientists, our colleagues from Clariant and Luna Vantage, who are co-registrants on the SI material in REACH, were also critical, and the consultants at ERM, Bibra and Vitis, who allowed the sharing of confidential information in some cases, not to each other, but with an honest broker third party, were really critical and got over some of the logistic hurdles in terms of how do we actually generate these refined exposure levels. And finally, as I say, it's no surprise that we publish this work. So the references at the bottom there, I invite you to go and have a look at that paper. And with that, happy to take any questions.
Great, thanks, Adam and Steve. That last comment that you made about data sharing reminded me that this is a really good example, not only for the science, but sort of the practical parts of how do you advance this type of approach. It's not only the laboratory science, it's working together really across different companies and different areas of and expertises. So very interesting. Thanks for sharing. We have a number of questions in the Q&A. I see that there are some questions in the chat. Please, we can't address them in the chat because it's too much to go back and forth. So if you have a question, please put it in the Q&A module, and then we'll get started. So the first question is, you kind of alluded to this already a little bit, but what are the possible improvements that could be used to move away from assuming 100% systemic bioavailability via inhalation, which the questioner says is conservative, which might be fine, but then if you get a BER that indicates high risk, is it possible to refine that exposure, or are there other opportunities via ADME testing?
Yeah, it's a great question. Thanks, Jay. At the time when we were doing this, it was a pragmatic choice. We had two routes of exposure. We had a standard OECD test guideline available for dermal penetration. And obviously, it was a kind of a no-brainer, albeit a slightly expensive test. But to refine the internal exposure estimates, that was a no-brainer. There isn't an equivalent OECD test guideline for refining the inhalation dose. I think there are quite a number of in development, more experimental assays out there, and I'd really welcome any references or any insights that people have. But there wasn't that off-the-shelf solution. And of course, what we did was run with the conservative assumption of 100% and see where we netted out. And actually, with some refinement of the external exposure estimates, so switching from some of the external exposure models that just give wildly unrealistic inhalation or air concentrations to ones that do factor in the fact that this is a non-volatile chemical, etc., we were able to achieve favourable BERs. So just like any risk assessment, if you don't need to jump to the next stage, we didn't. And in this case, we could tolerate that uncertainty or over-conservatism in the exposure, the inhalation exposure, sorry.
Okay. Anything to add, Adam?
No, I was going to say I'm also very eager to see if anyone has any useful references for this area. I was just going to add, and some of the complexities from an inhalation perspective is obviously around within a factory, physchem properties can, the form that a substance is in can change from vapors through to dusts, etc., etc. So it is a challenge. Inhalation exposure isn't inhalation exposure. There's obviously lots of different forms, so it is a conceptual challenge as well as a bit of a method gap at the moment.
Yeah. So the next question is, has ECHA accepted your approach in this example?
I'd be disappointed if we didn't get that question.
Yeah.
Those of you that operate in Europe will know that it is extremely rare to get a rubber stamp of acceptance from ECHA. What you get is silence. So what I can say is that the deadline for the response to the compliance check for this particular substance, and correct me if I get it wrong, Adam, was March 2023. And we submitted an updated dossier, including all of the data that Adam has talked through. And actually, we did use another exposure-based adaptation as well as the NGRA. That was submitted, so what's that now? Almost 2.5 years ago. It passed the completeness, both automated and human checks, so the manual and the automated completeness checks, but you do not get an acceptance as such. But 2.5 years have passed, and we've not had any correspondence from ECHA formally on this dossier. Informally, again, people operating in Europe who've attended the roadmap workshops will know that the noises and the signals coming out of the European Chemicals Agency are much more progressive than they were 5 or even 3 years ago. So I think it's highly unlikely that they're going to come out and say, yeah, well done, Unilever. This is exactly what we wanted. But the environment is much more receptive. And it is the way that the science is going. So we are hopeful that even if they came back with some challenges on certain parts of it, that it wouldn't be a case of merely dismissing it. And we are confident from a legal and regulatory standpoint that we have complied with the requests. I won't get into all the boring legal jargon, but there are certain phrases you have to provide substantial new information that's not manifestly unreasonable, that is related to the information requirements. We did that. The dossiers that we put in were massive, as you can imagine, with all of the experiments and the data that Adam has talked through. So I don't think anyone can accuse us of shirking or trying to take the easy path on this. Quite the opposite.
Okay. Next question is, how is maternal toxicity taken into account in NGRA as opposed to classical DART studies?
Yep, so it's a good question. So obviously, within the toolbox that we've developed, we've got a couple of tools for developmental and reproductive toxicity. They're based more around their validation, or their kind of the methodological journey that they've been on is more around the prediction of teratogenicity rather than maternal toxicity. We're not necessarily trying to predict specific effects like maternal toxicity, and instead we're trying to identify thresholds, generic thresholds for some biological changes that would be protective of maternal toxicity. So by using things like a single gene level change in a transcriptomic study, you are essentially being protective of maternal toxicity and therefore and any other kind of particular apical toxicities.
I guess the only thing to add is in terms of the PBK modeling, again, so Adam mentioned that there's a population simulation element of that. So both pregnant and non-pregnant populations were simulated from gestational age 1 week to 36 weeks, I believe, and very conservative assumptions around placental transfer and fetal concentrations were included. So again, building in lots of conservatism on that exposure side.
Yep.
Cool, great, thank you. Okay, there's a question about different types of substances. So if I have a substance with more activities and more complex modes of action, can I still select the same in vitro bioactivity studies to determine the point of departure? Great question.
That is a very good question. So I mentioned, so within the toolbox evaluation that we've done, it's mainly not too driven by hypotheses that you might have a priori. It's mainly driven as a generic first phase approach. We've got several cases internally on business relevant chemicals where we are paying a lot of scrutiny as to whether they are appropriate to be used in these bioactivity studies. So we are doing systematic literature reviews, very, very broad, much even broader in silico profiling than we have done for SI, for example, given this was a couple of years ago now, to really actually scrutinize whether those tools and approaches are appropriate or not. So we've got some examples running internally around actually having some studies that will lead in to whether we actually run the full toolbox based on priority modes of action, priority effects that we think potentially our toolbox might not be protective for because we haven't yet tested hundreds of chemicals. So we are placing a lot of emphasis into making sure in the real world use, we understand whether a substance we think is going to be adequate to actually run in that toolbox. But in terms of, if you've got a substance that's more active or not, then as Steve mentioned, the workflow that we've come up with has got plenty of opportunities for refinement, particularly in the exposure estimation area. So so in the past, we've gone to higher tier occupational exposure models, but potentially you could go to more refined PBPK models, etc. So just because SI didn't show much bioactivity, we have used much more potent occupational exposure relevant substances as well. So yeah.
Okay, great. Thank you. Let's see, one more question. You mentioned the TIMES platform for some of your work. Could you briefly explain what the TIMES model is and what you see as strengths and weaknesses in the context of your analyses?
Love it, because that harks back to my computational chemistry days.
That's your dream question, Steve.
So thank you for the question. So the TIMES platform is an in silico suite of predictive tools for various toxicological and ecotoxicological endpoints. One of the strengths of TIMES is that it implicitly includes a metabolism simulator as well. So you get predictions not just for the parent structure that you put into the software, but also the probable metabolites. In this case, as I say, lots of different endpoints in there. In this case, I believe we used the TIMES genotoxicity endpoint predictions as part of a broader suite of carcinogenicity and genotoxicity predictions. So we used Derek Nexus, OECD QSAR Toolbox, Vega, and TIMES to give a broad coverage of endpoints that our toxicologists would say, if you see a flag for these, that is something that you might then want to investigate further from a systemic or DART perspective. So it wasn't just carcinogenicity and genotoxicity. We looked at endocrine-mediated effects, models that predict DART toxicity, either apical endpoints or molecular initiating events. even more general screening like protein binding, DNA binding type alerts. So it was a very broad brush in silico profiling exercise, in the same way that the in vitro bioactivity measurements were also broad brush. But yes, the particular strength of Times is the integration of metabolism. So you do get that parent and metabolite prediction level.
Okay, great. Thank you. Okay, another question came in. So it says it's looking to know how long it took for you to take this process. And I guess maybe they're expecting or kind of realizing this is a pretty resource intensive approach. So do you have any comments about kind of cost and time effectiveness?
Yes, and I can spin this two ways depending on who I'm trying to convince. But in terms of cold hard cash, significantly cheaper than the study that was being requested. So one of the studies requested was a extended 1-gen repro study, and I'll get it wrong, but they're not cheap. Probably somewhere between 500,000 and 1.5 million euros, depending on the cohorts and that kind of stuff, right? So major cash. The cost for the testing that Adam talked you through was significantly lower than that. But of course, I also showed you the long list of names and the expertise, which I'm conscious is not available to anyone, to everyone, sorry. So where we stand today, there's a big effort to try to democratize a lot of this science, hence the publications, hence the presentations. Vast majority of the experimental work wasn't done at Unilever, it was done at CROs. Some of the consultants that we briefly flashed up are also now very well versed in NGRA, and we know and we're glad that they're actually helping our suppliers to conduct NGRAs completely independent of Unilever. So like all new methods, there is a bit of a hurdle to get over in terms of expertise, I would say, more than cost and availability of methods. But it certainly has the potential, because of the tiered nature, it has the potential to get through vast swathes of compounds for a much lower cost with a higher degree of protection. I don't think it's too bold to say that. Adam, anything to add?
No, I think that was very nicely put, Steve. I think currently the gap, I would say, is in the external availability of consultancy services and groups that can not just run the studies but pull it all together in the context of a risk assessment as well. But as Steve said, we've been working with several consultancy firms that I think now are able to operate fairly independently in terms of advising consortia and other clients with this kind of approach, and have a bit of confidence in the use of this data, and hopefully the more case studies that come out in this area just slowly builds up the availability and the ease for people, really.
Yeah.
Which I think goes some way to answering the next question that came about making the toolbox accessible. It's published, the names of the CROs and things are all in there. They're probably authors in some of the papers. And I can say even things like PBPK, which is quite a specialized endeavor. Whilst we use some commercial software, there's freeware available. And as time's gone on, I'm not sure if it's a good thing or not, but some of our scientists at Unilever obviously move on to new challenges, and some of those are at PBPK companies, and I know are being used as consultants for some of our suppliers. So again, the pool of people that are aware of these things and have the skill sets is growing all of the time.
Yep. I think the only currently internal piece of the puzzle that isn't commercially available is our pipelines and our dose response analysis that we use for transcriptomics, but we have plans to make that available in the very near future. And of course, there's a myriad of potential alternatives in the analysis of transcriptomics data. So I don't think that's a particular thing that's holding the field back.
Yeah, I think that's one of the areas. I think there's a lot of discussions in the literature and around with a lot of different scientists. So it's different ways to set that teapot and
Yep.
what makes the most sense. So, well, we got through all of our questions. Thank you all of you for listening, and thank you for adding your questions. Thanks for engaging with the speakers. Many thanks to Adam and Steve for giving their presentations, sharing your work with us, and we'll see you at the next webinar. Have a good summer, everyone.
Thanks all. Great to talk
Thanks,
to you.
everyone.